The overarching theme of BIO202 is the narrative of all of life, and you should be reminding yourself of this frequently so you don't lose your way and end up wallowing in the mass of factual information in the absence of context. It's this context that makes the material all come together, and it also makes you a better biologist. Now, I've told you that the main diversification of prokaryotic taxa into the major clades we identified occurred during the Archaean eon between cellular origins and 2.5 billion years ago. I've given you some examples of organisms living today that belong to the prokaryotic clades we covered in the last lecture. But this isn't to say that E. coli or spirulina were actually around during the Archaean. These two are modern organisms, the latest renditions of long lineages of living cells, stretching back as distinct from other lineages all the way to the Archaean. Presumably, their Archaean ancestors would still be recognizable as proteobacteria or cyanobacteria based on the synapomorphies of the clades, but they would be different from the proteobacteria and cyanobacteria of today. The full range of diversity of prokaryotes in the Archaean will never be known. Bacteria and archaea don't leave fossils except under highly specialized conditions like stromatolites, and when they do leave signs of their existence, it's often just in the form of carbon with a low, that is, biogenically consistent, ratio of carbon-13, and on very rare occasion, a microscopic smudge on a rock that would tell us that some kind of cell was there. With such a sketchy fossil record, there's really no way to tell how thick with life the Archaean was and how diverse it was, except, of course, from what we can surmise from the enormous range of prokaryotic diversity that exists today. We can say with reasonable certainty that this life would have been using anaerobic forms of metabolism, as there was effectively no oxygen, O2, until the Archaean Proterozoic break. And this is significant. No O2 means there would be no spontaneous formation of strong oxidizing agents like peroxides or superoxides that are damaging to biological molecules like nucleic acids and proteins. Peroxides form as water interacts with O2, creating molecules like H2O2, hydrogen peroxide, the same chemical that you can buy in the drugstore in a brown bottle. The reason why H2O2 is a good disinfectant is that pathogens tend to be poorly protected against the oxidizing effects of peroxide. H2O2 breaks into two equal hydroxide free radicals, which are just one electron shy of a complete valence shell and are highly reactive. They rob an electron from any nearby electron source, and in a cellular context, that source would likely be a protein or nucleic acid. And thus peroxide kills bacteria by fatally altering their molecules. In lab, we saw how the enzyme catalase is a protection against the harmful effects of oxygen. It works by destroying peroxides as soon as they form, not giving them a chance to form free radicals and harm the cell. Throughout the Archaean, there would have been no need for cells to have catalase or any other kind of antioxidation shield. But at 2.5 billion years ago, the onslaught of cyanobacteria making use of oxygenic photosynthesis, remember this equation, meant that this was all going to change. Having an abundance of O2 meant the spontaneous formation of not only peroxides, but also other highly reactive things like superoxides, and this would have had seriously damaging consequences for living cells. As far as the Archaean life was concerned, the Great Oxygenation was a horrible catastrophe that unfolded over the period marked by the Archaean Proterozoic break, spanning from 2.5 to 1.9 billion years ago. This is the period in which we can see banded iron formations in oceanic sediments, corresponding with the periodic events of oxidation of dissolved iron. Eventually, most of the ocean's iron settled out in its oxidized forms, and it's also around that time when O2 would have become a permanent fixture in the atmospheric chemistry. For life, this was a major challenge. O2 would not have been tolerated by cells accustomed to the anaerobic conditions that had prevailed over the preceding billion years. There are some modern cells living under exclusively anaerobic conditions, some living in your gut, and these will die instantly upon contact with air. This shows you how dramatic the damaging effects of O2 is. Oxygen is presently at 21% of our atmosphere, and much higher than the low levels that prevailed during the Proterozoic. But still, just the presence of O2, even at a very low level, would have been highly problematic, if not deadly toxic, to the unprotected cells. With a change in atmospheric chemistry caused by the cyanobacteria, there came the need to evolve and adapt to this new O2-containing atmosphere. Catalase is just one example. Our cells have 6 or 7 antioxidation defenses, including catalase. Adaptation for some cells, including the lineages of archaea and bacteria, leading to the modern cells identified as strict anaerobes, simply meant restricting their existence to habitats that are permanently anaerobic. I mentioned your gut as one such habitat. The majority of cells living in the human gut is not E. coli or even enterococci. The bulk of your intestinal bacterial flora consists of strict anaerobes, and the complete study of the gut community has not been possible in the past because of the difficulty in isolating and culturing species from that anaerobic portion of the microbiome. Now, however, we can use the big data approach of metagenomics to assess the total species diversity in the microbiome, and we do this by sequencing all the DNA found in the gut. And what we're finding is pretty mind-boggling. Not only is there a huge diversity of unrecognized, undescribed species, the human gut microbiome varies strongly from person to person, and it seems to be both influenced by the diet and state of health of the host, as well as having a sizable influence on the host's physiology. Every year, we see more and more studies demonstrating that the microbiome is causal to different things. Immunity and obesity are two aspects of human health for which causation by the gut microbiome is relatively well established. There's a growing interest in fecal transplants for the establishment of healthy microbiomes as a medically sound therapy. But at this stage, it's not entirely clear what the mechanism is by which the microbiome composition affects the host, and thus it's not clear what it is about a microbiome that would make it promote better health in a sick patient. Also, you'd have to find a way to successfully move the diverse complement of anaerobes in order to have a chance to establish the donor's full microbiome in the recipient. Now getting back to the question of oxygen, when you look at those cells that don't need to live in anaerobic habitats, they have obviously evolved some protection, like catalase. But the other important innovation that's at play here is aerobic respiration, actually using the O2 and its sizable oxidation potential to drive electron transport for the production of ATP energy. Here is the standard reaction summarizing aerobic metabolism. Starting out with some kind of food molecule, here it's a sugar, C6H12O6. Oxygen basically comes in and rips all the electrons off the food molecule, leaving only CO2. The amount of energy released by this reaction is much, much greater than anything that could be achieved without the oxygen. Aerobic metabolism, that is, using oxygen in this way, would not have evolved prior to the availability of O2, and hence we place this chronologically after the evolution of oxygenic photosynthesis. And this is something we talked about in an earlier video. Bacteria and archaea of the time would have been under the same pressure to adapt to the new conditions, and many evolved aerobic metabolisms. The highly adaptable proteobacteria would have been among the first to do so, and it would have been one such aerobic member of the alpha proteobacteria that went into an anaerobic host cell and became the ancestor of all eukaryotic mitochondria. In summary, the great oxygenation event was caused by life, the cyanobacteria, and it had dramatic and largely devastating consequences for life. In the aftermath of the oxygenation, life on Earth had a new tool for acquiring energy from the food molecules, aerobic respiration. Don't forget that this same oxygenation event was an important causal factor for the longest global cooling event in the Earth's history, putting the whole planet under a layer of ice for hundreds of millions of years. I think it's worthwhile to point out here that life was the causal driver of these massively important changes to the Earth's biological as well as climatic landscape. This is a theme that we see again later, notably in the Carboniferous with the activity of land plants, and also presently in modern times with the activity of our own species.